Antibiotics. Antibiotics are also known as antibacterials or antimicrobials. These are medications that destroy or slow down the growth of bacteria. These are medicines which are specifically used to treat diseases caused by bacteria, whereas some of the antibiotics can also destroy protozoans. Sometimes the harmful bacteria become excessive in our body and our immune system cannot fight against them. Then antibiotics are useful in this scenario. Antibiotics work in two ways. They kill the disease producing bacteria by interfering in the formation of their cell wall or any of its cell contents, or the antibiotics stop the bacteria from multiplying. So antibiotics can be defined as carbon compounds, and these carbon compounds are obtained from some bacteria or fungus. And what is the purpose of these carbon compounds or antibiotics? They are used for destroying or preventing the growth of harmful microorganisms. So carbon compounds obtained from some bacteria and fungi for destroying or preventing the growth of harmful microorganisms are called as antibiotics. And how do the antibiotics work? We have already seen they work in two ways. They either kill the disease producing bacteria or they stop them from multiplying. The discovery of antibiotics has brought about a revolution in the field of medicine. Antibiotics can be broadly classified into two groups: broad spectrum antibiotics and narrow spectrum antibiotics. So let us see what are broad spectrum antibiotics. Broad spectrum antibiotics are used when a bacterial infection is suspected by the doctor, but the name of the bacteria is not known, or sometimes it happens that infection takes place with multiple bacteria. So the term broad spectrum antibiotics are useful against a wide variety of bacteria, that is when multiple bacteria are present into the blood, or when the bacteria is not identified even after the blood test, but the symptoms of the disease are still visible to the doctor. So broad spectrum antibiotics are used when the pathogens cannot be identified even though the symptoms of the disease in an individual are visible. Some of the examples of broad spectrum antibiotics are amoxicillin, levofloxacin, streptomycin, tetracycline, penicillin, ciprofloxacin, cephalexin. In contrast to broad spectrum antibiotics, the narrow spectrum antibiotic is effective against only a specific group of bacteria. So whenever the pathogenic microorganism is definitely known, then the antibiotics are known as the narrow spectrum antibiotics. That is, when a blood test is done and the pathogenic bacteria is detected in the blood, then the narrow spectrum antibiotics are prescribed by the doctor. Examples of narrow spectrum antibiotics are penicillin, gentamicin, erythromycin, vancomycin, etc. So what is the basic difference between broad spectrum and narrow spectrum antibiotics? Broad spectrum antibiotics are those antibiotics which are used when a bacterial infection is suspected but the bacteria is not known, or when the infection takes place by multiple number of bacteria. How is it known? It is detected in the blood test. On the contrary, narrow spectrum antibiotics are those antibiotics which are used when the pathogenic microorganism is specifically detected during the blood test. Additional information. The building that you can see on the left side of the two images that are there on the slide is the National Institute of Virology. It was established in Pune in the year 1952. This institute takes up research work which are related to diseases like measles, jaundice, fever and diseases of the lungs. The National Institute of Virology is also a monitoring center for diseases like influenza, Japanese encephalitis, the rotavirus, measles and hepatitis. It is designated by the WHO, that is the World Health Organization, as the H5 reference laboratory for the Southeast Asian regions. So basically NIV is an institute which undertakes researches related to viruses. Penicillin. Penicillins are a group of antibacterial drugs, and these drugs can attack a wide range of bacteria. That is, it can be used as a broad spectrum antibiotic for destroying a wide range of bacteria. The discovery and manufacture of penicillin has saved millions of lives. So it has become a boon for mankind. Sir Alexander Fleming, a Scottish researcher, is credited with the discovery of penicillin in 1928. Let us now see a YouTube video created by Daniel Gochlin to see how penicillin was accidentally discovered by Alexander Fleming.
How Alexander Fleming accidentally discovered penicillin. Alexander Fleming was born into a modest Scottish sheep farming family in 1881. He excelled in school and entered St Mary's Hospital in London to study medicine. He developed a reputation as a brilliant researcher. His chance discovery of penicillin in 1928 has become a legend. At the time, Fleming was doing research on microbes. He was a very untidy person, and in August he went on holiday without cleaning his laboratory first. As a result, he left plates of bacteria culture in the laboratory next to an open window, and they stayed there for 2 hot summer weeks. While he was away, fungus spores blown in from the street contaminated one of his culture plates, and when Fleming returned from his holiday, he noticed that a mold had grown on that plate. He was about to wash the plate when suddenly something else caught his eye, something truly extraordinary. The mold had destroyed all the bacteria that were around it. Fleming had just discovered, entirely by accident, the first antibiotic, and this discovery was destined to revolutionize medicine. Fleming immediately realized the importance of this antibacterial substance, and as the mold belonged to the Penicillium family of fungi, he named it penicillin. He investigated the effects of penicillin on the bacteria causing pneumonia, syphilis, meningitis, diphtheria, gangrene, and other deadly infections. He published his findings in 1929, but little attention was paid to the article. Moreover, his penicillin experiments encountered serious difficulties because it was difficult to mass-produce penicillin in sufficient quantities. Consequently, Fleming abandoned his research. But 11 years later, in 1940, 2 other brilliant scientists, Howard Florey and Ernst Chain, started new penicillin experiments and demonstrated the miraculous effects of penicillin injections. After the outbreak of World War II, the British government and US pharmaceutical companies started the mass production of penicillin in order to treat all the Allied forces. By the end of World War II, it had saved millions of lives, and the legend of Fleming, the hero, grew. In 1945, Fleming, Florey, and Chain were awarded the Nobel Prize for Medicine. Penicillin and antibiotics generally are now recognized as the most efficient, life-saving drug ever discovered.
So we have seen how penicillin was discovered by chance by Alexander Fleming when he was on a vacation. He had left the laboratory completely unorganized, and when he returned after 2 weeks, he found the staphylococcus plate was contaminated with the fungi or the mold, you can say. And when he examined the mold, he found that the mold prevented the growth of staphylococcus bacteria. Penicillin has reduced the number of deaths and amputations of troops during World War II. To date, penicillin has become the most widely used antibiotic in the world. The mold or the fungus from which penicillin is made is Penicillium chrysogenum. Penicillin has a wide range of applications in controlling the bacterial infections. Bacteria like the Staphylococcus, the Clostridium, the Streptococcus bacteria. Penicillin antibiotics are used to treat infections which are caused by these susceptible bacteria. As you can see here, penicillin is used for treatment of the ear. Eye infection is treated, the nose infection is treated, the throat infection is treated with penicillin. Penicillin is also used for treatment of pneumonia and scarlet fever. Skin infections, bacterial infections in the stomach, infection in the kidney, infection in the intestine are also treated with penicillin. Penicillin is used for treatment of bacterial meningitis in the brain. Are antibiotics given to human beings and animals the same? Now, antibiotics can act only against a specific group of microorganisms. We know that they are bacteria. And these microorganisms which are present in human beings and animals are the same. Therefore, majority of antibiotics which are given to human beings and animals are the same. If the disease-causing bacteria is the same in human beings and animals, then antibiotics are given in specific doses according to their respective weights. How are vaccines produced? So before understanding the production of vaccines, we must know what is a vaccine. A vaccine is a biological substance designed to protect human beings from infections caused by bacteria and viruses. For making vaccines, disease-causing microorganisms like bacteria and viruses are used. But these bacteria and viruses are either in the weakened form or they are in the dead form. The first step in generation of a vaccine is generation of an antigen. An antigen is any foreign substance which induces an immune response in the body by production of antibiotics. So, an antigen induces an immune response in the body. This step includes growth and harvesting of pathogen itself or generation of a recombinant protein, that is protein which is made with a DNA technology derived from that particular pathogen which causes the disease. Viruses are grown in cell cultures, whereas bacterial pathogens are grown in bioreactors using a growth medium developed to optimize the yield of antigen. The second step is to release the antigen from the cell and isolate or separate it from the material which is used in its growth. Proteins and other parts of the growth medium may still be present and therefore they must be removed in the next step. So the goal of this second stage is to release as much bacteria or virus as possible. The third step is purification of antigen. For vaccines that are made from recombinant proteins, the steps used are chromatography and ultrafiltration. The fourth step is addition of adjuvant, which is a material that enhances the body's immune response. Stabilizers are added to the vaccine to increase their shelf life, and preservatives to allow multi-dose vials to be used safely. The final step involves combination of all components that make up the final vaccine and uniformly mixes them into a single vessel. Then the vaccine is filled in vials or syringe and packed with sterile stoppers. In order to understand how vaccines are produced, please find a video attached from YouTube by Sanofi Pharmaceuticals.
It's a simple gesture, one we make dozens of times for ourselves and our families to help prevent potentially life-threatening infectious diseases such as meningitis, pertussis, flu, polio. Vaccines work by preparing the immune system to better combat infection and prevent disease. They are biological products that result from a series of sophisticated and careful scientific processes that ensure the quality of each shot. Here's how vaccines are made. Production starts with growing viruses or bacteria in the lab. A wide range of parameters must be managed, like temperature, pH, oxygen rate, sterility, and homogeneity. The slightest variation in the culture media may compromise the production and cause delays. Depending on the vaccine, cell cultures take from 2 days to 3 months. From a small amount of original cell culture solution, the manufacturer can eventually produce millions of vaccine doses. The microorganisms are extracted from their environment through a process called purification, which eliminates any trace of culture media. We then inactivate the virus or bacteria. That means we eliminate its ability to cause disease. We disarm it. But we retain its ability to elicit a precautionary immune response from the body. The inactivated virus or bacteria is called a valence. A vaccine can include one or several valences to protect against one or multiple diseases in the same shot. Each valence must remain fully effective, and it must not interfere with the other valences. This careful, precise combination takes years of scientific research and development to perfect. Then, stabilizers or a preservative are added in extremely small quantities to help guarantee that the vaccine will remain stable, potent, and effective. Sometimes an adjuvant is also added to enhance the body's immune response to the vaccine. Certain vaccines are also freeze-dried into a powder to help further preserve the vaccine's efficacy. This is particularly important for vaccines that are intended to prevent outbreak-prone diseases like yellow fever, where having available stocks on hand over time is an important element to combating and containing disease. When it's time for the vaccine to be administered, it will be reconstituted by combining this powder with a diluent. The vaccine in liquid or freeze-dried form and diluents as needed are then filled into vials or syringes. When the filling phase is complete, each vial or syringe undergoes visual inspection. The quality of the contents and the container are scrutinized by both the human eye and state-of-the-art digital surveillance technology. All along the production, Sanofi Pasteur performs hundreds of laboratory tests on each and every batch of vaccine to confirm quality. In addition, samples are taken from each batch and sent to public health authorities for testing as well. Only after the vaccines have successfully passed both sets of tests can distribution begin. There are no exceptions. If a batch does not meet the quality criteria, it will be destroyed. Throughout packaging and shipping, vaccines must be stored between 2 and 8 degrees Celsius. Cold rooms and temperature-controlled shipping solutions, such as refrigerated containers and isothermal packaging, protect the vaccines during each phase of transportation and storage. A temperature tracking device is included in each shipment. Sanofi Pasteur takes care to follow strict precautions throughout the entire manufacturing process to ensure the quality of our vaccines. Production takes place in absolutely sterile conditions. Even the atmosphere is purified. Continuous control along the production chain also ensures that raw materials, equipment and manufacturing processes, and the finished product are of the highest quality. In total, more than 70% of the time required to produce a vaccine is spent on these quality controls. Depending on the vaccine, we conduct up to 1,200 different tests throughout the process. For these reasons, the total time required to produce a vaccine ranges from 6 months for the flu vaccine, for example, to 36 months for combination vaccine.
Vaccine given to the infants. So these are a list of vaccines which are given to the infants right from the time when the baby is born to the time when the baby becomes 1 or maybe 2 years old. So the vaccines which are given to the infants are polio vaccine, the DTP vaccine, which is a triple vaccine, which is very effective against diphtheria, tetanus and pertussis, the chickenpox vaccine, hepatitis A and B vaccine, the BCG vaccine, which is given to protect the body against tuberculosis, the influenza vaccine, the MMR, which is again a triple vaccine to protect the body from measles, mumps and rubella. When vaccines are injected in the body immediately produces antibodies in order to fight against them. And then these antibodies will remain in our body, and in future, whenever there is any attack by the disease causing microorganisms, these antibodies which are produced when the vaccine is injected into the body will help in fighting with these disease producing bacteria or maybe the viruses. It is necessary to store the pathogens of a disease against which the vaccines are to be produced. Now, whenever the pathogens for creating a particular vaccine are to be stored, then they have to be stored very safely, because pathogens means they are harmful disease causing microorganisms like the bacteria, fungi, viruses, etc. Now we know that for making vaccines, elaborate processes are used. These are very expensive processes. So in the course of time, when these pathogens are released in air, then it might prove to be very dangerous, because they may spread various diseases rapidly before the vaccines are available. Caution to be taken while administering antibiotics. So what is the first precaution that you will keep in mind while administering antibiotics? That antibiotics should be taken only when the medicine is being prescribed by the doctor. Don't purchase any antibiotic from a medical store without doctor's prescription. We are not doctors, so we do not know which medicine will be suitable for us. So always buy medicine only with doctor's prescription. Many times when we suffer from some simple flu-like infections or maybe a common cold or maybe some throat infection, what we do is we have a box, a medical kit at home, and we pop in the tablets without asking or without confirming it from the doctor. So it's very dangerous. Don't consume any antibiotics on your own to treat any diseases. It may be some common diseases like throat infection, common cold or maybe flu. Even if you feel better before completing the prescribed course of antibiotics, you must still continue to take the medicines and complete the course. If you leave the course incomplete, the microorganisms may still be present inside your body, and when you stop the antibiotics, these microorganisms once again will start rapidly multiplying in your body. So always complete the course of antibiotics that is being given by the doctor. A very important word of caution: don't suggest the antibiotics that you have taken to others.